Hello and welcome to your Microbiology Bio 203 video lecture on observing organisms through a microscope. I'm Mr. Kennedy and I'll be your guide as we explore this topic together. We'll begin today by first examining the microscope. The microscope is a simple instrument that has very humble beginnings. A simple microscope has just one lens and at one time in our history looked literally like a rudimentary magnifying glass. This is a picture of the first microscope that was used by men like Robert Hooke and Anton von Leeuwenhoek. In order to use this microscope, a specimen had to be balanced on a pin, and then the viewer would examine that specimen through the tiny lens while holding the entire apparatus up to the sunlight. Microscopes have come a long ways since the first microscope. Light microscopy is a microscope that uses visible light to observe specimens. There are now a number of different types of light microscopes: the compound light microscope, darkfield, phase contrast, differential interference contrast, fluorescence, and confocal. This is a picture of a simple compound light microscope. It has an ocular lens, in this case 2, so it would be referred to as a binocular scope, a body tube which transmits the image from the objective lens to the ocular lens, an arm, 4 objective lenses, a stage, a condenser which focuses light through the specimen, a diaphragm which controls the amount of light entering the condenser, an illuminator which is just the light source, and coarse and fine focusing knobs. In a compound microscope, the image from the objective lens is magnified both by the objective lens and the ocular lens. In order to determine total magnification using a compound microscope, the two numbers must be multiplied. Compound light microscopes also possess a prism which flips images upside down and backwards as you view them through the ocular lenses. The image that you see from the prism is also referred to as a virtual image, since it is a reflection of what is coming through the objective lenses. Resolution is the ability of the lens to distinguish 2 points. A microscope with a resolving power of 0.4 nanometers can distinguish between 2 points at least 0.4 nanometers apart. Any closer, and the 2 points would appear as one. In order to improve resolution, shorter wavelengths of light will provide greater resolution or resolving power. The refractive index is a measure of the light bending ability of a medium. Light may refract after passing through a specimen to an extent that it does not pass through the objective lens. Lost light means poor viewing. Immersion oil is used to keep light from refracting. This is an example of what would happen both with and without immersion oil. Bright field illumination. In bright field illumination, dark objects are visible against a bright background. Light is reflected off the specimen and does not enter the objective lens. This is an illustration of how bright field illumination works. Darkfield microscopy. In darkfield microscopy, light objects are visible against a dark background. An opaque disc must be placed in the condenser in order to accomplish this. Only light reflected off of the specimen will enter the objective lens. This is an illustration of dark field microscopy. Phase contrast microscopy allows examination of living organisms and internal cell structures. It brings together two sets of light rays, direct rays and diffracted rays, to form an image. This is an example of phase contrast microscopy. Differential interference contrast microscopy is similar to phase contrast in that it uses two light beams and prisms to split light beams. It gives more contrast and color to the specimen. Fluorescence microscopy uses UV or short wavelength light. Fluorescence substances absorb the UV light and emit longer wavelength visible light back. Cells may be stained with fluorescent dyes or fluorochromes if they do not naturally fluoresce. Confocal microscopy. In confocal microscopy, cells are stained with fluorochrome dyes. Short wavelength blue light is used to excite the single plane of a specimen. Each plane in a specimen is illuminated and a three-dimensional image is constructed with a computer. Two-photon microscopy. In two-photon microscopy, cells are stained with fluorochrome dyes. Two photons of long wavelength red light are used to excite the dyes. This allows study of cells up to 1 millimeter deep. Scanning acoustic microscopy measures sound waves that are reflected back from a specimen. This is used to study cells that are attached to surfaces and has a resolving power of 1 micrometer. Electron microscopy uses electrons instead of light as an illumination source. The shorter wavelength of electrons gives greater resolution. It's used for images too small to be seen with light microscopes, such as viruses. The transmission electron microscope uses a beam of electrons that will pass through ultra-thin sections of a specimen, then through an electromagnetic lens. Then that image will be focused on a projector lens. Specimens may be stained with heavy metal salts for contrast. This is an illustration of a transmission electron microscope. Transmission electron microscopy can magnify objects 10 to 100,000 times and has a resolving power of 10 picometers. Scanning electron microscopy. Scanning electron microscopy uses an electron gun to produce a beam of electrons that scans the surface of an entire specimen. Secondary electrons emitted from the specimen produce a three-dimensional image. This is an illustration of a scanning electron microscope. Scanning electron microscopes can magnify objects 1,000 to 10,000 times and has a resolving power of 10 nanometers. The scanning tunneling microscope uses a tungsten probe to scan a specimen and reveal details of its surface. Has a resolving power of 1/100th of an atom. Atomic force microscopy uses a metal and diamond probe placed onto a specimen and can produce three-dimensional images. Preparation of specimens for light microscopy. In order to view specimens under a microscope, it is often necessary to prepare them as a smear and stain them. Staining accounts for organisms that are normally translucent and difficult to see. Coloring microorganisms with a dye can emphasize certain structures under a microscope. A smear is a thin film of a material containing microorganisms spread over a slide. Microorganisms are fixed or attached to the slide, which kills the microorganisms and makes it possible to store the slide indefinitely. Live and/or unstained specimens have little contrast with the surrounding medium. Live specimens are used to study cell behavior. Stains consist of a positive and negative ion, one of which is colored. the colored portion of the stain is called the chromophore. In a basic dye, the chromophore is a cation. In an acidic dye, the chromophore is an anion. Staining the background instead of the cell is called negative staining. Simple stains. Simple stains use a single basic dye or a single basic stain and highlight the entire microorganism to visualize cell shapes and structures. Occasionally, a mordant may be used to hold the stain or coat the specimen to enlarge it. Differential stains are used to distinguish between bacteria. Gram staining and acid-fast staining are examples of differential stains. Gram stain. Gram stains can be used to classify bacteria into gram-positive or gram-negative groups. Gram-positive bacteria have thick peptidoglycan cell walls. Gram-negative bacteria have thin peptidoglycan cell walls and a layer of lipopolysaccharides. This is an illustration of the gram staining procedures. And this is an illustration of properly gram-stained cells. The cells that are purple in appearance are the gram-positive cells. The cells that are pink in appearance are gram-negative. Acid-fast stains. Acid-fast stains bind only to bacteria that have a waxy material in their cell walls, which is not decolorized by acid alcohol. Acid-fast staining is used for the identification of Mycobacterium endocardia. This table illustrates the color of acid-fast and the color of non-acid-fast cells as we move through the staining process. And this is an example of acid-fast bacteria. Special stains. Special stains are used to distinguish parts of microorganisms. Special stains include capsule stains, endospore stains, and flagellar stains. Capsules are a gelatinous covering that do not accept most dyes. Suspensions of India ink or nigrosin contrast the background with the capsule, which appears as a halo around the cell. This is an example of a capsule stain. Endospore stains. Endospores are resistant, dormant structures inside some cells that cannot be stained by ordinary methods. The primary stain in endospore staining is malachite green. It's usually with heat and driven into the cell. Endospore staining decolorizes cells with water, and then a counterstain is applied in the form of safranin. The spores will appear green with red or pink cells. This is an endospore stain. Finally is the flagellar stain. The flagellar stain is utilized in organisms that possess flagella for locomotion. It requires the use of a mordant and the primary stain, carbofusion, in order to visualize these structures. This is a flagellar stain. And this concludes our coverage of viewing organisms with a microscope.